Okay, every cell has a life cycle, and that life cycle we can kind of divide up into the cell's normal life being what the cell is, carrying on its normal metabolic processes, and that's called interphase. But then if a cell is triggered to where it needs to divide, needs to become two cells from one cell, then the cell will have a growth period, and it will have stages that actually prepare it for division. We talked about those in lab, but we may talk a little bit more about what actually goes on in that time before the cell actually divides. Now, when a cell divides, it actually kind of does this through both mitosis, what's going on with the actual DNA and chromosomes, and cytokinesis, which is where the cell actually splits in two. And of course, as cells do this, they do divide, so we can produce more cells, or so they can have a repair process. Now, not every cell divides after it reaches maturity. Every cell has the potential to divide at least sometime in its life cycle, but there are some cells that continually divide all the time. We continually need to replace them. There are other cells that once they're mature, there is no more division in them. And so we tend to see them lose their centrioles if they don't need to divide anymore. Whereas some cells, such as certain skin cells, we have what we call stem cells that are constantly going through cell division. And then we have other cells that they divide to a certain point in their life cycle, then they stop. But then there may be something such as an injury, like a broken bone or something like that, that would trigger cell division to occur again to help repair this. Whenever it is determined that a cell needs to divide, one thing that has to happen is we're going to have to replicate the DNA, and so when we do divide the cell into two cells, each new daughter cell has the same amount of chromosomes, the same amount of DNA material that the original cell had. So part of this occurs at the end of interphase, preparing for mitosis. And what's actually happening is we actually have an enzyme called DNA polymerase, and it starts unzipping the DNA from two different sides in actuality, and it starts unzipping those hydrogen bonds between the nitrogenous bases and attracting the DNA nucleotides that are complementary to the existing bases. So notice we have something called an old template strand, and the old template strand was from the original DNA. The new template strand is what we're forming. The enzymes are actually attracting the nucleotides, and they are forming bonds to the existing strand. So when we're finished with the process, we have two identical strands of DNA. Therefore, we can actually wind up and form chromosomes that we can pull apart. So when we do divide into two cells, we have the same DNA in both of the cells. Again, the cell division is really divided up into two parts: mitosis, what's going on in the nucleus, what's going on with the DNA in the nucleus, and the formation of the two new nuclei for the two new cells that are forming. And we have cytokinesis, which is what's really going on with the rest of the cytoplasm. And this doesn't actually happen until we get to around the telophase stage of mitosis, but we then do divide one big cell into two, what we call daughter cells. Here we see a representation, interphase being the normal life cycle of the cell, but growth, DNA replication, even replication of the organelles occurring once the cell is triggered that it needs to divide. We know we're in early prophase once we see the chromatin is wound up into the chromosomes. We see the chromosomes are made up of two identical sister chromatids that are attached by a centromere. We see that our nuclear envelope, that double membrane envelope, starts fragmenting, and that our centrioles start moving to opposite ends of the cell, and they drag their spindle fibers across the cell, and the spindle fibers actually attach to the centromere, and as the centrioles move to opposite ends of the cell. So we know that we then go on to our next. Notice that the metaphase is where our chromosomes have actually been dragged across the middle, or what's called the metaphase plate of the cell, and they're lining up across the middle, and then that phase is interrupted as soon as the spindle fibers start pulling the sister chromatids away from each other, and as soon as the centromeres are divided and one sister chromatid starts going one way, the other goes the other way, we're in anaphase, and they continue to be pulled towards the centrioles that are on opposite sides. Now finally, we get to telophase, where we actually start seeing a cleavage form in the large cell to start dividing it into two cells, our cytokinesis, dividing up our cytoplasm and our organelles. We also see that our chromosomes that have been pulled to opposite sides of the cells, we start forming a nuclear membrane around them and forming two opposite nuclei. Our cleavage furrow continues until you can still see remnants of the spindle fibers where the connections are, but the cleavage furrow keeps continuing until we actually have a separate membrane between each cell and therefore separate daughter cells. We know that genes are important. We know they have a lot to do with what determines our characteristics, but what a gene really is basically a segment of the DNA that is a blueprint that tells us how to build one protein or polypeptide. And then we know that proteins, actually, that's the real determiner of our characteristics. The many different proteins that have different shapes, do different jobs, work with other proteins to do different jobs. So most of our building materials for our cells, such as whether it be something like hemoglobin, very large protein, whether it be keratin, whether it be actin, myosin, troponin, tropomyosin, a lot of different proteins serve as building materials that do different jobs for the cells, and even for outside the cells. And then we know that enzymes, which are, again, our biological catalysts, they actually help us put things together chemically, they help us break things apart or rearrange things. All our enzymes are proteins, and without these, then we're not going to have the metabolic chemical reactions, therefore life's going to cease to exist pretty quickly. And then, of course, RNA is actually, we have three different types, and all three types are used in the building of proteins, as we will see. Again, there are three different types of RNA, and we will look at these three different types. Notice we know that the DNA carries the blueprint for RNA, so how are we going to get the information from the DNA to the factory or the ribosome where we actually build the protein? Well, we're going to have a special type of RNA called messenger RNA that's going to carry that instruction from inside the nucleus to the actual ribosome. We'll be talking about that in great detail. We know that ribosome, where we build our proteins, our factory, is made up of basically proteins and this type of RNA called ribosomal RNA. So to build a protein, we have to have a protein, but we also have to have the ribosomal RNA to make up our ribosome. And then finally, we have to have what we call transfer RNA, because we've got to bring our building materials to the factory. And of course, our building materials, our foundation of the protein, is our amino acids. So something has to be able to bring the appropriate amino acid and read the instructions that are found on our messenger RNA, and this is going to be transfer RNA. All this we're going to cover in a topic called protein synthesis, which is made up of something called transcription and translation. Okay, transcription, basically, if you transcribe something, you're writing down information. So what we're going to do, we're going to write down information from our blueprint on DNA. We have what we call a template strand and a coding strand. And so we're going to copy the template strand. We're going to form something called codons, which is going to be the foundation of our messenger RNA. And that's all going to occur inside the nucleus. Next, we're going to go through our next step of protein synthesis, which is translation. And that's basically where we take our messenger RNA, we take it to a ribosome, and then we bring transfer RNA and we read the messenger RNA with the transfer RNA, and we put together these amino acids that help us make our proteins. That's going to call translation. That's occurring at a ribosome, which is outside of our nucleus.